This lesson's about the ultrastructure, or the fine-level structure of cells, the subcellular anatomy. These are your objectives. So before we get to the ultrastructure of cells, let's talk about the different kinds of cells. So we can classify cells into two general groupings, prokaryotes and eukaryotes. And the karyote part refers to the nucleus. It actually means kernel and is used for the nucleus, whereas pro means before, and eu, which is a prefix we'll see a few times in biology, means true. So eukaryotes are things with a true nucleus, prokaryotes come before the nucleus. So prokaryotes can be divided into two groups, and it's a bit misleading to do it the way I'm doing it here, because archaea are actually more closely related to eukaryotes, but the way I'm dividing it, the archaea and bacteria are both kinds of prokaryotes, and neither of these have nuclei. So they have no nucleus, and along with that, they have no other membrane-bound organelles. In other words, they're not compartmentalized internally by membranes. You can say they're not compartmentalized, or they have no membrane-bound organelles. Means the same thing. Whereas eukaryotes do have a nucleus, and they do have membrane-bound organelles. They are compartmentalized. The membranes inside are dividing them into different regions. A nice analogy I've heard of this is that you can think of prokaryotes as being like a camping tent, where everything inside is in one space, whereas eukaryotes are more like a large house, where there are different rooms and different things can be happening in those different rooms without interfering with each other. You can have someone playing the piano in one room and someone eating in another, and they're not going to interfere. You could have someone talking on the phone, etc. So different rooms with different things going on, different compartments bound by membranes. Let's unpack this a little bit further. So prokaryotes are simpler and smaller than eukaryotes. Because they're not compartmentalized, they can't have as many different things going on in different regions. When we say they're smaller, they're sort of on the order of about 1 to 5 micrometers. A micrometer is a millionth of a meter. A millimeter is a 1/1000th. This is 1/1000th of that. So 1 to 5 millionths of a meter. And again, they come in these two kinds. Archaea, which are what we often think of as extremophiles, things that live in like hot water vents underwater, hot springs, high salt environments, and bacteria, which are sometimes called eubacteria, to distinguish them from archaebacteria, which is another name for archaea. Okay, so archaea and bacteria, both prokaryotes, no nuclei, no compartmentalization. Eukaryotes are larger, and so they're on the order of sort of 10 to 100 micrometers. And they are what we think of when we are talking about plants, animals, fungi, etc. These three together, archaea, bacteria, and eukaryotes, are the three domains of life. And then once you get into the domain, for example, in eukaryotes, you can talk about kingdoms, Plantae, Animalia, etc. Okay, so those are the three domains of life. Now, another thing about prokaryotes divide by binary fission. That's how they reproduce, because they don't have a nucleus that needs to be divided, whereas eukaryotes have to divide the nucleus to reproduce. And so they either do that by mitosis, if they are preserving the number of chromosomes, or they do that by meiosis if they are having the number of chromosomes in preparation for sexual reproduction. And we'll talk about both of these more in the course. So that's prokaryotes and eukaryotes. Now, before we look at the ultrastructure, let's talk about how we can see it. Let's talk about microscopes. So we can classify microscopes into light microscopes and electron microscopes, named because light shines a beam of light at an object, and electron microscopes, instead of using light, are shining a beam of electrons. Now, if we start with electron microscopes, the advantage of that is that they have higher resolution. And resolution, higher resolution than light microscopes. Resolution is the ability to separate or distinguish two objects from each other. So it's not only a matter of magnification. You could magnify something really, really big, but if you magnify it and it's fuzzy, that's no good to you. Resolution is the ability to separate two objects from each other, and that's actually really what counts most when we're talking about microscopy. So electron microscopes allow higher resolution, allow us to separate two things. So instead of seeing something like a blob that looks like that, maybe, you can see it as two things like so. You can separate the two of them from each other. Electron microscopes allow higher resolution, and their resolution is about on the order of a little bit more than 1 nanometer. So they can separate things at that level. Whereas light microscopes have a resolution that is more like 1 micrometer. So again, a micrometer is a millionth of a meter, a nanometer is a billionth of a meter. Now the advantage, however, of light microscopes is that you can study living cells, which you can't do because of the preparations required with electron microscopes. Just for comparison, by the way, the eye has a resolution of about a millimeter. These are sort of order of magnitude estimates, but you can see that it goes up by about 1,000 times as a kind of rough way of remembering it. And that's also illustrated over here on the diagram where you can see the unaided eye, light microscopy, and electron microscopy. Notice that you can see some eggs, including maybe just barely a human egg with the unaided eye. You can see cells, plant and animal cells, and you can see the existence of bacteria and mitochondria with light microscopy, but you can't see well inside them until you get down into this region using an electron microscope. And you can't see viruses and even the existence of ribosomes, the subcellular protein-making machinery, without an electron microscope or using other more fancy kinds of light microscopy. Okay, let's take a quick tour of prokaryotic and eukaryotic cells. So first, here's a prokaryotic cell. You've got an electron micrograph and then a corresponding diagram. A few things to notice. There's no nucleus. There's a nucleoid region. The nucleoid region is a region where the DNA is, but again, it's not membrane bound. There's both a cell membrane or a plasma membrane and a cell wall. So this cell membrane is internal to the cell wall. The cell wall is outside, a protective structure. Sometimes students get confused and think that there's one or the other, that when there's a cell wall, there's no plasma membrane. There's both here. There can also sometimes be stuff outside the cell wall, like a capsule of jelly-like material, also protective. You can see here there are these long whip-like structures. These are called flagella, and they help it get around. And there can also be fimbriae, which are attachment structures. You'll also sometimes see structures called pili, that's plural, the singular is pilus, and that's an extension that comes out from the bacterium that is used to transfer bacteria back, transfer DNA back and forth between bacteria in a process called conjugation. Lastly, just note what we do and don't see here in terms of subcellular organelles. We do see ribosomes. Bacteria have ribosomes, but they don't have membrane-bound organelles. They don't have mitochondria, chloroplasts, etc. Ribosomes are not membrane-bound. Bacteria have ribosomes. Prokaryotes have ribosomes. This is another diagram from Campbell biology. Now we're looking at eukaryotic cells. So very quickly, notice that we've got a nucleus, which is made of a nuclear envelope with stuff inside it. That's where the DNA is kept in a eukaryotic cell. Continuous with the nuclear membrane is the endoplasmic reticulum. This is another membrane-bound organelle, and it's involved in synthesizing membrane and proteins. It's divided into two parts, the rough endoplasmic reticulum that has ribosomes on it and is involved in making proteins, and the smooth endoplasmic reticulum, which doesn't and has other functions. Eukaryotes can have flagella too. Down here we see the Golgi apparatus. The Golgi apparatus is also involved in protein processing for export, another membrane-bound organelle that we didn't see in the prokaryotes. Similarly, we didn't see mitochondria, or as we'll see when we look at plant cells in a moment, this is an animal cell, chloroplasts in prokaryotes. They're all membrane-bound organelles. But once again, we do see ribosomes. Ribosomes are in both pro and eukaryotes. They're different in the two. That's just a quick tour. You'll need to take a longer look at this to really internalize the concepts. Let's look at plant cells now. Here's plant cells again showing us these components, the endomembrane system, which is the nuclear envelope, the endoplasmic reticulum, and the Golgi apparatus forming part of that. You see a nucleus because it's a eukaryote. You see ribosomes, which are in both pro and eukaryotes, mitochondria, and chloroplasts. Chloroplasts now because it's a plant cell. Sometimes students get confused and think that plant cells have chloroplasts but not mitochondria. They have mitochondria as well. We'll explore further in the reading and the practice what each of these do. Lastly, just distinguishing plant cells from animal cells. Among other things, plant cells have a cell wall. Of course, just like before with prokaryotes, they have a plasma membrane internal to that. The cell wall of plants is made of different material, cellulose, than that of prokaryotes. But it also provides structure, and they have a large central vacuole which is involved in providing for its structure and can be involved in storage as well. So that's some differences between plant cells and animal cells in addition to the presence of chloroplasts. So there's an introduction to prokaryotes and eukaryotes and the ultrastructure of cells.